Welcome back, class. We are going to be talking about the neuroglial cells or the neuroglial cells now. So remember, within the neural system, we have two types of cells. There's the neuron, which we talked about in a previous video, where we talked about the structural and functional classification of a neuron from a structural point. They were multipolar neurons, they were bipolar neurons, and there are unipolar neurons. Give me 1 second, just want to make sure. There we go. Again, from a structural perspective, there are multipolar, bipolar, and unipolar neurons. And then from a functional classification, we said that there are somatic or sensory neurons, I should say, motor neurons, and association neurons or interneurons. Now, when we talk about the other type of cell, remember neurons are also electrically excitable. The other type of cell are called neuroglial cells, again, meaning nerve glue. And they make up half the volume of the central neural system. They're much smaller than neurons. So if we're looking at this picture off to the right, the large thing in the center, that's a neuron. And everything that we see around it would be neuroglial cells. They're 50 times more numerous, those neuroglial cells. And we'll focus on four types that are located in the central neural system. And then we have two types that are located in the peripheral neural system. That's why it's important to differentiate CNS from PNS, central neural system, from peripheral neural system. The four types in the central neural sites, oligodendrocytes, microglial cells, and ependymal cells. And then the two in the peripheral neural system are Schwann cells and satellite cells. And of more importance, we'll focus on the Schwann cells. So this is just looking at a general overview of so many different types of terms that we see. So up here on the top, these are cells of the Piamata. Cells of the Piamata is a layer of protection around the brain and the spinal cord. That's the Piamata, right? You've heard of the term meninges. I have a video on the spinal cord and a video on the brain that goes into the meninges. You've heard of meningitis. And there's the Piamata, the arachnoid, and the duramata, these three layers, Piamata, arachnoid, and duramata. So this is the Piamata. And then we see some cells like an oligodendrocyte, a microglial cell, astrocytes, ependymal cells. We've heard of myelin sheath, node of Ranvier, right? These are neurons. These are ependymal cells down here on the bottom, which we'll get into, that have microvilli and cilia coming off of them. So let's look at the first cell, the astrocyte. Within the word astrocyte, we see astro, meaning star or star-shaped. Astrocyte, astrology. The astrocytes are important because they're going to form the BBB, the blood-brain barrier. That's the astrocyte. It forms the blood-brain barrier. It's the connection between the circulatory and the brain and the spinal cord. And it's involved in helping the body metabolize neurotransmitters. It's involved in regulating potassium balance. Remember, potassium is important because of the sodium potassium pump, and we need that for depolarization and repolarization, depolarization and repolarization. So when we talk about action potentials, that's very important. The oligodendrocytes. Okay, we've seen the term, but we don't know what it is yet, and it is analogous to the Schwann cell. So remember, the Schwann cell myelinates in the peripheral neural system. Oligodendrocytes are going to myelinate or form myelin within the central neural system. Okay, so the oligodendrocytes are going to myelinate in the CNS, whereas the Schwann cells are going to myelinate in the PNS. What is the disease associated with the breakdown of that myelin sheath? That is what we call multiple sclerosis. Multiple sclerosis is an autoimmune condition where it attacks the oligodendrocytes or the Schwann cells. And we need the insulation around these axons to speed up conduction and make sure that the electrical current is flowing beautifully. In terms of multiple sclerosis, the first part of the body that is typically affected is going to be the eye, because the nerves and the neurons of the eye are the smallest. So the eyes get affected first, and typically there's visual disturbances when there is demyelination of the optic nerve, which is cranial nerve number 2 is the optic nerve. Okay. But the good news is that the optic nerve and cranial nerves are part of the peripheral neural system. And when we have damage to the PNS, the neural system can repair itself. When there's damage within the CNS, that's where damage becomes more permanent. Peripheral nerves and myelin within the peripheral neural system can repair and regenerate. When it's outside of the brain and spinal cord, it's easier. When it's within the brain and spinal cord, the damage is much, much harder to fix. So here is what myelination looks like. So we see that we have the axon, which is the yellow in the middle. Here is a Schwann cell, and you can see that the Schwann cell with the nucleus is engulfing the axon, and it just kind of makes a circle around it. It's going to keep making a circle, till eventually we have this nice layer of insulation wrapping all around it. That's the myelin sheath. Okay, and the node of Ranvier is that gap in the space between the next Schwann cell. Okay, microglial cells, these are going to clear microbes and cellular debris. So they're phagocytic. They clear away dead cells. They're derived from monocytes. All right, so monocytes are a type of white blood cell, and monocytes are always in circulation. They're always within your blood vessels. Monocytes can evolve into a macrophage. Macrophage, phagocytic. See the phage? See the phage? They're cell eating. They engulf. So monocytes become macrophages. Macrophages are found in tissues. So monocytes are in blood circulation. When the monocyte finds damaged tissue, they move into the tissue and now they become a macrophage. When the monocyte moves into the neural system, it's called a microglial cell. When it moves into the lungs, it's called an alveolar dust cell. When it moves into the liver, it's called a Kupffer cell. When it moves and engulfs damaged cholesterol, like really oxidized LDL cholesterol, then it's called a foam cell. So monocytes are a type of white blood cell. It's an agranular a granular site, a granular, meaning there's no granulations in the cytoplasm. Kind of has a cell, and then it's got this really, really big nucleus that's almost the same size, like this. Sometimes it looks kidney bean shaped, so let's make it a little bit better. So let's say you got this large cell with this large nucleus that looks like that, like a kidney bean shaped. So that's a monocyte, okay? That's a monocyte. If you have another cell, forget about that. If you have a cell with a nucleus that's really spherical and round, and again, there's no granules in the cytoplasm, there's no granules in the cytoplasm. When that nucleus is large and pretty much as large as the cell, that's your lymphocyte. So this is your lymphocyte, that's your monocyte. They're agranular. There's no granulations in the cytoplasm. Okay. When we get more into white blood cells, when we do circulatory system, we'll go over the agranular sites and we'll go over the granular sites, which have granules in the cytoplasm. All right, ependymal cells. Ependymal cells are really important. You can see they have cilia there, and they produce CSF. CSF stands for cerebral spinal fluid. Cerebrospinal fluid. They're found in these cavities called ventricles. And within the brain, we have this ventricle system where we have um, lateral ventricles 1 and 2, and then they are going to come down into ventricle 3, and then eventually into ventricle 4. Okay, so you have cerebral spinal fluid flowing up here. This is all in the brain, by the way. This is all within the brain. I'll show you shortly. We have these lateral ventricles, one in each hemisphere of the brain, and it's going to dump cerebral spinal fluid into the third ventricle and dump it again into the fourth ventricle. And these are drainage ducts. Think of them as that. This is called the foramen of Monroe, and this is called the cerebral aqueduct. Foramen of Monroe, sometimes called the interventricular foramen of Monroe, and then you have the cerebral aqueduct. All right, so all around these are the circles, think of them as ventricles, and you're going to have cells that line these ventricles. The cells that line them are ependymal cells. The ependymal cells are producing that cerebral spinal fluid. The purpose of the cerebral spinal fluid is to help bathe and protect the brain and the spinal cord. Okay, and it's also involved in bringing electrolytes and glucose, sodium and potassium exchange through the neural system. Satellite cells, not too much important stuff there. They are just supporting neurons. That's really all they are. They're supporting neurons of the PNS. Okay, and the other important cell of the PNS, that is the Schwann cell. Again, much like the oligodendrocyte, the Schwann cell produces myelin in the PNS, the oligodendrocyte produces myelin in the CNS. So again, you see that it insulates, surrounds the axon, keeps going around and around and around and around, and all of this stuff here going around it, this is all yellow, all of that is the myelin sheath. Now what's interesting is that On the left-hand side, I showed you this in a previous video, where everything on the inside, the letter H, right, that's gray matter, and everything around it is white matter, which means everything outside of it is myelinated, okay? But in the brain, it does a flip-flop, right? In the brain, the gray matter is on the outside. The outer part of the brain is called the cortex, right? So the cortex of it is gray and the inside is white. So what you see in the spinal cord and what you see in the brain in terms of white matter and gray matter, they're opposites. They flip-flop. In the spinal cord, the butterfly or the H is gray, white matter is on the outside, but in the brain, the gray matter is on the outside, myelinated white matter on the inside. So now when we look at this is up before on some of the Connect, McGraw-Hill Connect quizzes. So what we see is the letter H in the middle, spinal cord. That is gray matter. That's myelinated. Now, we could go as far as identifying dorsal horns, dorsal roots, ventral roots, because what we're seeing here, this is the vertebra, right? So this is the vertebral body. The discs, the intervertebral discs would be here, which means everything on this side is anterior. This is the spinous process of the vertebrae. So that means back here is posterior or dorsal, and this is ventral. So if we look at these nerves, right, this one here, that's a dorsal root, and then this on the front side, that's a ventral root. So this dorsal root, that's sensory. The ventral root is motor. Gray matter is the orange, white matter would be around it. Then I'll put a little dot here. That's your dorsal horn, and that's your ventral horn. All right, so now what is the highlighted orange structure here? Well, that's going to be white matter. Okay, good place to take a break. When we come back, we'll get into the brain and some of the primary functions of the brain.